So when I talk about autosomal dominant conditions, your hereditary spherocytosis, your achondroplasia, all these are autosomal dominant conditions. When I talk about autosomal recessive conditions, your cystic fibrosis, your hemochromatosis, Gilbert syndrome, all these are autosomal recessive conditions. X-linked recessive conditions is your color blindness, chronic granulomatous disease, your Duchenne's, muscular dystrophy, G6PD deficiency, hemophilia. Okay, then you have Rett syndrome, vitamin D resistant rickets. All these are X-linked dominant. Now for mitochondrial, you have to remember that you get mitochondria only from your mother. So whatever mitochondrial genes are there, which help in synthesizing your proteins, which are enzymes or proteins for the electron transport, the respiratory chain, are all maternally inherited. Okay, so mitochondrial disease will follow a maternally inherited pattern. Examples of mitochondrial disorder, very important MCQ, Leber's, Mela's, Merck, Kearns-Sayre, and sensory neural hearing loss. Okay, apart from that, there is a fragile X syndrome, which comes under trinucleotide repeat disorders, which we are going to see. Okay, sometimes I put these slides because directly ask you what is the inheritance of AIP. They may tell you patient is coming with neuropathy, pain in abdomen, okay, sub-psychological symptoms, patient symptoms got worse after taking sulfa drugs, or patient is on ocipils and the pain is increased. What is the inheritance of this? So that time you have to understand that they're talking about intermittent porphyria and accordingly answer the listing. Okay, now anticipation is a phenomena which is seen in trinucleotide repeat disorders. Okay, these are nothing but a repetitive sequence of 3 nucleotides that get amplified with each generation. So suppose in generation 1 has 10 nucleotides or 35 nucleotides is normal, 10 to 35. The next generation, which is having abnormal number of repeats, okay, is having 60. The next generation after that will have 80. So as the generations increase, the number of nucleotide repeats increases as compared to the normal. So that there are two features that occur with successive generations, that the disease comes earlier and is more severe. This is called as a phenomena of anticipation, which is seen in trinucleotide repeat disorders. Repeats found in which disorder, which repeat, like Huntington is CAG, Friedreich's is GA, Fragile X is CGG. This is all important. Okay, and the exception to this, not showing anticipation phenomena, is your Friedreich's ataxia. Okay, important is Huntington's is your CAG repeat, Friedreich's is GAA, and Fragile is CGG. Okay, next there is genomic imprinting. We know that there are two alleles of a disease, of a gene. Okay, one is maternally inherited, one is paternally inherited, and therefore we always write it like that when we write about our pedigrees. Out of this, one could be in silence, or one could be inactivated, and the other may be functioning responsibly, and that could be either from your paternal listing or maternal inheritance. Okay, disease arises when this is also normally inactivated, but the paternal or the maternal functioning gene also gets silenced. Okay, this is the concept of your genomic imprinting. Okay, so paternally silencing gene is Prader-Willi. Mothers are angels, maternally implanted, Angelman, P. Prader-Willi, P. paternal. Prader-Willi, you have to remember, as a cause of genetic obesity, whereas Angelman is usually ataxia, seizures, and developmental delay. Okay, so your Dijot syndrome is a microdeletion syndrome, catch-22, you have to remember. Okay, so we saw the DNA replication, we saw the DNA mutations that can occur, the eukaryotic transcription steps. This is the protein synthesis, the polymerase chain reaction. In this, you just have to remember the steps: denaturation, annealing, and elongation. Okay, these are the 3 steps. Basically, what it does, if you see here, it amplifies your DNA so that it becomes easier to quantify, easier to recognize, easier to see which part is actually getting involved because of which disease. Okay, apart from that, your genetics, there's certain break to your cell cycle so that your cells doesn't continuously proliferate, multiply, and go mitosis wise. That is a tumor suppressor gene. So suppressor doesn't let cells to proliferate and form a tumor. Proto-oncogene, pro-proliferation. So more of oncogenetic, more of proliferation, more of genesis of cancer. Okay, so these are certain mutations and the genes which are responsible for that. Like we know, HFP was hemochromatosis, NF1 is neurofibromatosis. right? WT1 was WIMS, fibrillin or FBN1 is Marfan syndrome. Okay, so again, like I told you for AIP, they can give you these features and then tell you what is the dominant inheritance. Suppose I say cafe ole macules, axillary freckling, Lish nodules, plexiform neurofibromas, and then I ask you the inheritance, autosomal dominant, right? Suppose I tell you someone having recurrent sinopulmonary infections, fat-soluble vitamin deficiencies, malabsorption, growth failure, pancreatitis, diabetes, cystic fibrosis, autosomal recessive, Marfan's with these features, then you have to say autosomal dominant. Okay, so this is the part where there is genetics, which are important, the important concepts of genetics, which we have to be discussing.